Good day, my dear students. I would like you people to kindly visualize this class completely. It is on instruments and equipment as spotters. They may be asked in the Viva also. There is more to light than the opposite of dark. I had chosen opposites as the theme of my class. Look at this beautiful picture. Obviously, you people can appreciate the opposites. It is left to you to find out what all it contains. Thanks to this beautiful reference. I am finding a picture of a needle over here, which is strong and stout. It has got a lateral adjustable guard. Depending on the thickness of the area it is going to penetrate, it can be adjusted. And this incidentally is a stillet which can be removed, a syringe attached to it, and the aspirate made. So these are two parts, just remember it. With a guard, I would like to call it a Sala's needle. This you find that the lateral guard is missing. This is the climb. Either of the needles are short, stout, with a stillet, silver in appearance, and firm in consistency. They are bone marrow aspiration needles. Absolute indications for bone marrow aspiration will be aplastic anemia, megaloblastic anemia, multiple myeloma, and ITP. These four conditions alone you can remember for the time being. This is another needle. This is also a little long and firm and stout. So this is a stillet, and you find it is branching so that it can catch hold of the tissue, and when it is being withdrawn, you find the biopsy is there with us. It is the Jansi biopsy needle, firm, a branch stillet, and a bone marrow biopsy. Again, it can be used for aplastic anemia, metastasis, etc. So there are two things over here. In one, I find this kind of a needle. In another one, it is like this. Can you people appreciate the differences? Look at the wonderful picture on the screen. This has been created by an artist. I would like you people to understand the opposites. So what is the procedure that is going on over here? You find that the patient is in the left lateral position, a couched manner, and then there is a needle that is penetrating the intervertebral space to reach the subarachnoid, and then a sample of the cerebrospinal fluid is being obtained. Between the third and fourth is convention. I am not going into the procedure as such. CSF examination, normally the fluid is collected in three containers for biochemistry, microbiology, and pathology. Microbiology, of course, it has to be perfectly sterile for culture and sensitivity. Biochemistry, the various components of it. And in pathology, the type of smear or sometimes malignant cells. So these can be some indications for cerebrospinal fluid examination. Spinal anesthesia, incidentally, is a therapeutic use of lumbar puncture. This is a beautiful picture. You find that it is long, slender, and somewhat malleable when compared with the bone marrow needles. This is the picture over here. A lumbar puncture needle. And the complications can be infection, herniation of the brainstem if too much of aspirate is being made. So this is the reference for the picture that I showed. Now, this is a tube. I find that it is short and stout. It is closed over here and probably it is open at this end. Open at one end, 11 centimeters long, graduated for about 10 centimeters. And there are two types of graduation, 0 to 10 and 10 to 0, depending on the usage. So this is called a hematocrit tube or the Winthrop's tube. Hematocrit literally means packed cell volume. This is a counterpart of it called the Westergren's tube. It is a long tube. It measures about 300 millimeters. Graduations are from 0 to 200 and it is open at both ends. And this is the sample where the blood is collected and it is placed in it so that the blood goes up by capillary action. This is a Westergren's tube with the stand. It has to stand in a perfectly vertical position for a period of about 1 hour. You find that the diameter can be 2.5 millimeters and it has got a 1 millimeter hole. It is placed in an airtight screw on either side. And the ratio of the blood to the anticoagulant will be 1:4, 3.8% trisodium citrate as anticoagulant that is used over here. Now, this is the Westergren's tube and the Winthrop's tube. I hope you people can appreciate the differences. Again, the differences are mentioned over here. What do you infer from this tube? This is supposed to be the Westergren's tube, the Winthrop's tube or the PCV tube. Winthrop is used for the ESR and PCV for the packed cell volume. After centrifugation for a period of about 30 minutes, I find out that the blood is being separated into the plasma, the Buffy coat, and the packed red cells. What is the use of this particular one? From this itself, what can I infer? Plasma. In iron deficiency, it is pale. Jaundice, yellow. Cholesterolemia, it is waxy. And hemolysis, it is pink. So these are the changes in this zone. Buffy coat. Thin in a case of leukopenia, thick in a case of leukemia, and it can be made to diagnose subleukemic leukemia. Organisms such as microfilaria and cells such as LE cells may be found in this zone. And coming to the RBCs, it is increased in polycythemia and decreased in anemia. So these will be the uses. Have it in mind. So this is yet another spotter that we can have. It belongs to histopathology. Let us see what is there. What is the shape of these? L. So they are called as the L pieces or L molds, and the person who has designed it has got his name. So they are called as Leucart's L pieces. So they are used for molding the wax. You find that they can be kept side by side so that a perfect rectangular trough is being made. The advantage with the L is the shape and the size can be adjusted depending on the size of the tissue. It is usually kept on a flat or a glass surface. And the wax tissue blocks are prepared for having the sections cut. Look at this. This is a L and other L. They have been placed like a rectangle. The tissue will be placed in it and the wax is put. This is again a modern innovation. Almost the same thing. The wax is being poured into it. The tissue will be in it and it is again taken off after it solidifies. So there is a change in. This is a Leucart's L piece. See the tissue, the wax, and the square or the rectangle it forms. Finally, I have a wax block. This also can be a spotter for you people. A wax block is kept, you'll have to mention, it contains a histological section and then it is surrounded by paraffin wax. It is used for cutting the sections in a microscope. And after cutting the section, This is a slide that we people usually get. Look at the block over here and the corresponding slide. The shape is exactly maintained. Opposites. Wonderful reference. And I do not know from where they get such innovative ideas. One is the moon, other is the sun. Now, this is a diamond pencil. Supposed to be very costly. A pencil like thing, and it is really a very crude diamond. Diamonds are very expensive because of the fine sharpening and the facets that they obtain. But these diamonds are used for marking the glass slides. They mark a permanent edge on the glass so that they are not erased or lost. So, in order to maintain the histopathological number, you find that the diamond pencil makes us the mark or the number. Finally, it is being cut with the equipment called the microtome. It is quite sturdy, it can't be carried, it is not a spotter for you, but the wax block is kept here, knife is over here, sections are being cut. So this is the wax block we have been talking about. So this is a closer view of the microtome. Your block is here and ribbon-like sections are being cut. See this. This is what any histopathology lab will be getting. They get so many tissues and blocks are being prepared and multiple slides. If the mark has to be permanent, this writing labels will be dangerous. That is why we need a diamond pencil for making a permanent mark, avoiding mixing of tissues. Yet another one. There is a jar in front of me. Is there any particular feature? Leave alone my description. I am finding that there are multiple ridges over here. So there are 1, 2, 3, 4 ridges, which mean how many compartments are there? There will be 5 compartments. And this is a tightly fitting lid that can be placed over it. It can be used for histopathological processing or for staining. So another diagrammatic view of the same. See what is there. The ridges and then a tightly fitting lid. Sometimes it can be in the form of plastic also, but it has to be a material that is not melted by the reagents that are being used. This is looking at it from the top. I find that the ridges are there very clearly. Now, the advantage of this is I can place 2 slides in a ridge back to back in arrangement. So the opposite sides will be there. The sections will be opposite to each other. So 10 slides can be placed and at the same time the staining can be going on simultaneously. So this is the slide we should have in mind. So this is called the vertical Coplin jar, previous. Look at this fellow, a corollary of it. It is horizontal and there are multiple ridges and similarly there is a tightly fitting lid. It can be used for processing or staining. So this is called a horizontal Coplin jar. So world of opposites. I hope you people can make out the drawing on the screen thanks to R.A.T.S. We go to another set of instruments and this is a pipette obviously, which is bulged, which is called the bulb and it has got a red bead within it. Also, there is a rubber tube for drawing the sample and it has got a red colored mouthpiece. But what is important will be the markings 0.5, 1 and 101. The red bead should not be the answer. These should be the answers. Instantly, you find that the red bead is used for 1. Identification, 2. To know whether the pipette is wet or dry, and 3. for mixing the sample. These are the three functions of the beam. Its counterpart, the WBC pipette. So here again there is a bulb, but then what is important will be the markings 0.5, 1 and 11. These are the things. It has got a conical one, all the other parts are given over here. I would like you people to go through. Similarly, don't write the white mouthpiece alone as the answer. Graduation should be the first point, other things can follow. So the contrast between them we shall be seeing. This is a hemocytometer which encompasses the RBC pipette, the WBC pipette, and then there is a chamber over here, the counting chamber, which is also an equipment or an instrument for us. So this is the counting chamber. These are the cover slips. And when you put it under the magnifying lens, this is what we people see. There are 4 corner large blue squares which are each divided into 16 squares. Whereas in the middle, there is a larger one. I am finding there are 25 squares, and the corner squares and the middle one of these are used for RBC count. So this we can be mentioned. So the counting chambers, etc. This is the improved new bar chamber. Please make that answer. And what are the parts of it? You find that there can be the ridges. So these are all elevated. They are called the ridges. And then there is a trough for a hollow region, which is used for the outflow of the excess fluid. There is a central platform which will be having the counting ridges or chambers. Improved new bar chamber. That should be the answer. Another one, again in histopathology, can be the microtome knife. This may or may not be kept, but basically we should understand. A knife means, so this is a knife, the yellow arrow. The star is supposed to be the back, which supports it. And the orange arrow is the handle that is used for holding the knife. It is used for cutting sections in a microtome. But today, we are used to disposable blades such as this. This might be kept. So, see here, there is a blade holder and this is affixed to the microtome. I have already shown you that equipment. So, sections can be cut with it. So, it is a disposable blade, thin in nature, made up of alloy, fixed to the blade holder over here, used for cutting microtome sections. So it's the same thing. It's a holder, there's a blade over here, there's a microtome. Art science and art complete are complete opposites of each other. What are we seeing on the screen? I am finding a cylindrical glass tube. And there is also another friend of ours which has got a cylinder, and then there is a graduated tube. There is a bulb over here. The stem of it is graduated. Here I am not finding any graduation. Earlier mercury was used. Now an alternative is being used because of the toxicity of mercury. See, it is suspended in the urine, and when it floats, the lower meniscus is taken as the reading for the specific gravity. Urinometer used for specific gravity, you can mention the parts of it. So there is a bulb, and then there is a tube, and then there is a stem of it. Lower meniscus is taken for the reading. So it is suspended in a spinning motion without touching lower meniscus. Look at the opposite over here again. Courtesy R.A.T.S. There is another tube over here. In this tube, however, I am finding some graduations. And there is a container for this tube. What is this particular one? C. It is a tube in which the urine is collected, and then after some time, the protein sediments, and you can take a reading of the protein. So it is assess the proteinuria in a patient. This is called an S-Bax albuminometer. So you fix it, fill it up to the U mark, the urine and the reagent up to R. It contains picric acid, that is why we get this yellow color. It's a preservative because it has to stand for 24 hours. The name of the instrument is S-Bax albuminometer. So there's a urinometer. Sometimes reagent strips are also being used. You will have to read up the entire class of mine in proteinuria. Opposites. Again, a beautiful painting, HC. What have I tried to convey to you people? How do I remember so many instruments? Bone marrow needle, opposite of lumbar puncture. Stout, thin. There is a horizontal Coplin jar, vertical Coplin jar. RBC pipette, WBC pipette. L mold and a recent modification. Winthrop's tube, short and stout, Westergrens, long tube open at both ends. Microtop knife with a handle, etc. Disposable blade made up of alloy. Urinometer for measuring the specific gravity. Albuminometer for measuring the proteinuria. Gracias.